My name is Alice, and I'll be presenting on one of my summer research projects done at the UBC MRI Research Center. This center primarily focuses on applying advanced MRI techniques to the application of understanding MS, or multiple sclerosis pathology. And the major finding of this project was finding a negative correlation between the biomarkers that measure myelin content and water content in MS brain. So for me, what's the most interesting about MRI is to be able to quantitatively assess these microscopic biocomponents of the human body. So I hope that by the end of my presentation, you'll be able to understand why this finding is important in the context of MS research. I'll be talking about what exactly is MS, some background on MRI, and then explaining how these advanced MRI techniques can aid the understanding of MS pathology. Then I'll be going over the details of the experiment and conclude with the implications of our findings and the impacts of these findings on future projects. So to start off, who has heard of multiple sclerosis before or knows someone with MS? For me, I know 2 people personally with MS, and unfortunately Canada has one of the highest rates of MS in the world, so MS research is quite big in Canada. If you look on the picture here, both of these patients have MS. However, the person on the left could have the disease for a longer period of time, and we can clearly see that the person on the right will never walk again. So what MS is a neurodegenerative disease, and what that means is that the nervous system will break down over time. Unfortunately, there are no drugs that are able to completely stop the disease, and there is no chronological timeline where a doctor can tell a patient and say you have 5 more months to walk, or you have a year left to walk. Because the nervous system is breaking down, it can break down through various forms of pathology, and currently when you go into the hospital to get a scan, the doctor cannot tell you how the nervous system is breaking down. So what we strive to do is using advanced MRI techniques, we try to distinguish between the different pathologies. MRI is able to measure high quality images of inside the human body. It's able to produce these high resolution images because it relies on the exploitation of the physical properties of water and fat, which are the 2 most abundant biocomponents of our body. More specifically, it's actually imaging the hydrogen atoms in the hydrocarbon and the H2O molecules. Hydrogen atoms in water and fat give rise to differing intensities. That means the hydrogen atom signals can tell us information about water content inside the body and water environment. So as we can see here from these 3 scans, the different intensities can clearly distinguish between different tissue pools. So for example, there's the corpus callosum. My finger's not long enough. And to go more specifically into the technique of MRI, it is based on the idea of nuclear magnetic resonance, which is NMR, and with NMR, you are able to distinguish between chemical and physical properties of molecules. So from NMR signals that we receive from tissues, we categorize them into voxels, and voxels are representative of a volume of approximately 2 millimeters cubed, so you can kind of imagine that in your head. The MRI machine then makes pictures from these voxels that you see on the computer. And the intensity of the pixel corresponds to the intensity of the NMR signal per voxel. The brighter the intensity of the pixel corresponds to a higher intensity in the NMR of the voxel. And what I have shown here on the right-hand side, or your left, is the inside of the machine. It is composed of what we call gradient coils, and this controls the central magnetic field. On the right, I have a radio frequency coil, and this is used to send radio frequency pulses at the biological tissue, and the signal that we receive back from the tissue from the RF pulses is what we're reading from the machine. So 2 types of coils. And now I'll be going over the details of the specific advanced MRI techniques that we use for this experiment. One technique called T1 relaxation occurs when you send the radio frequency pulse at the molecules, we're imaging hydrogen, and when that happens, the hydrogen atom molecules will grow to a higher energy state, and this causes them to spin off the axis of the central magnetic field. When you turn off the radio frequency pulse, the hydrogen atoms will go back to the equilibrium state because that's the lowest energy, and that state happens to be when all the hydrogen atoms' macroscopic magnetization is aligned with the magnetic field. So the time it takes for realignment to occur is the T1 relaxation, and this time is dependent on water content. So the higher the water content of a tissue, the longer the T1 relaxation. The next technique that we use is called T2 relaxation. And what this measures is the transverse magnetization. I'll go into that further. So when the RF pulse hits a hydrogen molecule, it'll start spinning, and it'll go to a higher energy state. What that causes to happen is the magnetization of the hydrogen atom flips, And in the flipping, you create a transverse magnetization. The T2 realization measures the time for all the hydrogen molecules to dephase from each other. So to give you an analogy of how this works is if you imagine each hydrogen atom as a small clock, when the RF pulse hits it, they'll be spinning at the same amount of time, going the same rate. After I turn off the RF pulse, which you can program, after some time, they'll start spinning out of sync with each other. And the time it takes to go all out of sync is the T2 relaxation. What determines this is how tightly the water compartments are confined. So the less tightly the water molecules are, the longer the T2 relaxation time is, and the more tightly the molecules are, the shorter the T2 time. From the T2 time distribution, we can distinguish between 3 specific pools of water inside the human brain. The lowest peak is myelin water, because that is the water confined between the myelin sheath, and hence the pool with the greatest constraint. In the middle, we have intra-extracellular water, and at the end, we have CSF, cerebral spinal fluid, which is water associated with fats, lots of space. And from the T2 distribution, we can extract a value, which our lab discovered and made pretty much, called the myelin water fraction, and this is defined to be the area of the myelin water over the total area of the distribution. So why are these 2 advanced MRI techniques important to MS? When you go into the clinical scanner, this is a conventional image that you are able to get, and those scanners are very good at detecting lesionous regions, which are these bright spots. However, as the lesions grow bigger in MS, they can begin to look like tumors. So conventional MRI scans in the hospitals are unable to detect specific pathologies. And however, if you look at the white matter that appears normal, in MS we call it normal appearing white matter, advanced MRI studies have actually shown abnormalities. They show higher T1 relaxation times and lower myelin water fraction. So the purpose of this experiment is to use advanced MRI techniques and determine whether myelin water fraction and T1 presents any correlation. What we did was we scanned 18 MS subjects with relapse remitting MS and 18 age and gender matched controls. One of the subjects were on disease-modifying therapy. The scans were required on the Philips 3T Achieva scanner, and 4 scans were required every time. 3D T2 was used to acquire myelin water fraction. T1 immersion recovery is used to acquire water content. T1 relaxation. 3D T1 is used to define white matter, and FLAIR is used to define lesions. If anyone wants to know the specific parameters, please talk to me after the presentation. To look at only the normal appearing white matter, we created normal appearing white matter masks. We did that by subtracting the lesion area shown in blue from the total white matter shown in green. So what we were left with are the red mask here. For the data analysis, the T2 relaxation time distribution was calculated for every voxel using a modified extended phase graph algorithm combined with a regularized NALS flip angle and optimization. And the T1 distribution was calculated again for every voxel using a model exponential fit. If anyone would like to know the mathematics behind it, please speak to me after the presentation. We plotted histograms for all the voxels for every single subject and extracted mean, peak height, and peak location. Then we compared the mean, myelin water fraction, and T1 for every subject using a linear regression. And the results that we found was that we found a significant negative correlation for every subject for every month. And this is important because it shows that there is a correlation between these biomarkers. What was even more interesting was when we plotted the data for the controls on, we still found the negative correlation with the R-square of 0.57 and a p-value of less than 0.001. So the biological significance of this finding is that a physiological relationship may exist between these 2 biomarkers that measure myelin content and water content. And more significantly, that means there's probably a biological linkage between demyelination in MS and increasing water content. However, this is just a correlation, so we can't say this is causation. So further studies need to be done. And what we're doing now is we're looking at the lesion of myelin water fraction versus T1. As well, we can also look at post-mortem brain slices and measure the demyelination using histological techniques and compare that to water content. So to bring it back to the greater picture of MS research, what does this mean? This means if we are able to prove biologically that demyelination is causing an increase in water content, then we will be able to diagnose patients in the future according to where they lie on the regression scale. And then to look further ahead in the future, drug development companies can develop drugs based on where a specific patient lies on a cohort of the scale. So that's the end of my talk, and I would like to thank my PI and associates in the MRI research section. Thank you. That's a person with a lot of lesions. So if you remember the white focal spot that I showed, that's a person with a lot of lesions that look like tumors. And we actually checked our data by